Welcome back, students. This is the first video in a sequence of modules on Chapter 8. Chapter 8 deals with how genes are expressed. Now, as we learned in the past, gene expression is simply the production of something from the information contained in that particular region of the DNA. This could be a protein, or it could be an RNA molecule that has a particular function. An important commodity for students to consider in this chapter and in the next chapter. Questions such as these produce in the students an understanding of that particular concept. So here we have a scenario where more than 90% of the genes encoded by the mammalian genome produce proteins, and at the same time only 10% of the genes produce RNA. So here we are looking at the number of genes. One could falsely assume that means that 90% of the product inside a cell from gene expression are going to be proteins, and only 10% of the total volume of product is going to be RNA. That is not the case. Looking at the answer to question A, we find that RNA represents only 3 to 5% of the volume of product, while the vast majority is going to be protein. One finding that amazed scientists when first discovered is that in a multicellular organism like plants and animals, we have different types of cells tasked with different types of functions. And we assumed in the past that genes would be deleted so that only the genes necessary for that particular cell would be maintained. This turns out not to be the case. All cells, with a few exceptions, inside an organism contain the entire set of DNA that they inherited from their mother cells. So what makes different cells different from each other? The answer is simple. The amount of DNA stays the same, but the activity of that DNA is varied. This is very nicely illustrated in this figure here. These two cells were taken from the same individual, so they contain exactly the same DNA inside their nucleus. However, you can see the morphology of the neuron is completely different to the morphology of the white blood cell that you see as a dot down in the corner here. So what makes the two cells behave and have different anatomy and physiology? The answer is that they express different genes. Physiologists tell us that the human body has over 200 different types of cells. Each cell can be distinguished from another cell type by its pattern of DNA expression. So even though, once again, these two cells have the same identical DNA in their nucleus, they do have very different functions. So regulated gene expression means that genes are controlled in some sort of sophisticated way that allows these cells to have different morphologies. So diversity between cells from the same DNA is driven by differential gene expression. Evidence for these findings came from both natural and artificial experiments conducted by humans. So here we have proof, more recent proof, that a nucleus contains all the DNA necessary for making a brand new individual, in this case a frog. So what we have done here is we've taken skin cells from an adult frog, mature skin cells, which are highly differentiated. We remove the nucleus, only the nucleus from those cells, and that nucleus is then placed into another cell, an egg cell, which has had its nucleus removed or destroyed by UV light. So this enucleated cell, and that's the word that we use for a cell that has no nucleus, is then given a nucleus from a skin cell. Under the right conditions, this cell can be made to differentiate into a tadpole, and the tadpole will then eventually form an adult frog. So this proves that the information carried in this nucleus here is enough to direct this cytoplasm to make a fully differentiated frog with all the different tissues having all the appropriate functionality. More appropriately, experiments on plants have been performed for about 8,000 years. So we see that we can take a cell from any part of a plant and grow that into a brand new plant by a vegetative cutting. I'm sure your grandmother or your mother have been known to take cuttings from a plant that they liked at a relative's house, bring that cutting back, put it in soil, water it, and that grows into a brand new plant. So that's the principle behind here. The difference between this and the previous slide is that here we're taking whole cells or whole tissue and then encouraging that to grow into a fully grown adult plant. Slightly different. The application of what we have learned from experiments such as the frog is amazing. So now around us we have animals which are important to our society that are produced by cloning. So a farmer has a cow that produces the best milk in the herd, and that cow is now getting old, so the farmer wants to make sure that in the future all cows produce milk at the same level. So the technology now exists and incidentally is used by local farms where cells from the udder of this cow or any other part of this cow can be taken, and just like the frog example, their nuclei are removed and they are replaced calves, which then mature into adult cows. When it comes to understanding which sets of genes are shared by different cells and which ones are unique to a particular cell type, this slide helps us understand that type of compartmentation. So housekeeping genes are genes which are needed by all cells in that organism to survive. And examples include common things that take care of common elements within a cell. So histone genes produce histone proteins, and all eukaryotes have the need for histone proteins. So all cells in eukaryotes express histone genes. Basic metabolism, like glycolysis, that's shared by all cells in an organism and in multiple organisms. So those genes are also turned on and kept on throughout the life of those cells. And of course, we have other genes that we'll talk about in other chapters that are necessary to be kept on at all times. So those are called housekeeping genes. Those genes are necessary for the survival of those cells. The other class are the specialist genes. These are turned on only when necessary in particular types of cells. So here we have an example of a brain cell. It will have only its genes turned on at the appropriate time, whereas a liver cell will never turn on brain genes unless there's a mistake. Likewise, kidney cells. So these are called specialized genes or specialist genes, and they are turned on only in that particular cell type. So this chapter addresses how that control can be manifest. Going on to this slide, this just tells us that most cells, as we mentioned at the beginning of a course, respond to their environment. So the environment is the main driver behind turning genes on and off. And as we said, later in this chapter and the subsequent chapter, we'll learn how this is done. But it's very important to understand that cells respond to their environment, therefore taking action by turning on, in most cases, certain genes and shutting down other genes, right? And then finally, this is the last figure that you need to understand for this video. This summarizes a consensus of examining thousands and thousands of genes. So scientists have looked at thousands and thousands of genes in different animals and different creatures, down to bacteria, and they've classified them according to the level at which that gene expression is controlled. So for instance, it could be that in gene A, the gene is controlled at this level. So the gene is only turned on when needed. Otherwise, the gene is shut down. But that's not always the case. It may be the majority, but in some other cases, we find that genes, their products are controlled at a slightly different level in their expression. So some genes are controlled at the RNA processing level. And you can see number 3, that happens a bit further out in the cytosol. Number 4 is a way of controlling which RNAs are kept and which ones are removed. Control level 5, which happens in other genes, there the translation on the ribosome is then regulated. And number 6, the proteins themselves can be removed from the cell when not needed by a process of protein degradation. And number 7 simply says proteins can be turned on and off rapidly depending on their need. More about this later, but the essential thing that students have to take away from this figure is that most genes are controlled at level number 1, but there are other genes that are controlled at these other different control points. Incidentally, it takes about 4 hours for something to be turned on at number 1 before the protein appears in the cytoplasm. So that's considered a slow response. It takes a long time, and the cell may not have that time to respond to its external clues. So in those cases, those proteins may be regulated at the end of the pathway, where the response can be rapid, within seconds, within milliseconds, a cell can respond. You'll get to understand this concept as we move forward in this chapter. For now, thank you.